Now, there are 4 models that we use. Yes, there's 5 things up there because the last is us. But we don't use humans as a model organism because the model organism, what it's modeling, is us. We really, when we use vertebrates for study and development, again, it's about us. It's about learning how humans develop without actually having to do experiment and genetic studies on humans. A few people have done that in history, and they're not very nice people. Xenopus, fish. So Xenopus is the frog. Fish is the zebrafish, which the species is Dirario. Chick we call Gallus is the species. Gallus gallus is the domesticated chick or chicken. And then mouse, Mus musculus, is the genus species for that. And then Homo sapiens are humans. So these are the 4 vertebrate model organisms that I'm going to talk about: Xenopus, fish, chick, and mice. So the first thing we're going to talk about is the advantages and the disadvantages, just like we talked about before with the invertebrate models, on why we use them for various purposes, and then, of course, what we learn from each one of them. So that's really what we're about. We're answering the human questions of development. So let's start with the Xenopus. Here are the advantages. The cells are so large you can actually see a frog embryo with the naked eye. Now, you can see a tiny little dot, but most cells you can't even see with the naked eye. So because the cells are large, we can see a lot of the micromeres and the smaller cells that develop as a result of that. That's one advantage, is the cells are so large we can look and watch the development of these in a great amount of detail. Frogs, in their earliest stages when they become tadpoles, have very rapid development. And obviously, it slows down after that, and then they'll go through a metamorphosis into the adult stage. But in the initial stages, when they first develop into the tadpole, it's very rapid. So the more rapid turnover of an organism, the more we can study its overall development, which is a great advantage. We use frogs primarily for transplantation experiments. So the moving tissues from one area to another to answer the questions of specification and commitment of these cells. This is where we've learned quite a bit about that. Now, we can use other organisms for this as well, but frogs in particular are more advantageous for these transplantation experiments. Now, unlike Drosophila, which are great for genetic studies, Xenopus are not very great for genetic studies because, like us, they have multiple chromosomes. And it gets very difficult to show one-to-one relationships between mutagenesis of a particular gene and the resulting developmental defects that come from that. Drosophila and some of these invertebrate model organisms are great for genetic studies because of that one-to-one relationship between a mutation in the gene and the development of certain regions. But frogs start to get more complex, like us, in the multiple chromosomes, as well as the way in which they regulate their genetics, as we know, as far as gene splicing and things of that sort. Another disadvantage is you can't go through many generations with the frogs because they go through a long growth period before they're fertile again. So unlike Drosophila and some of these other organisms where we can keep breeding them generation after generation and look at the long-term effects of mutagenesis and other genetic studies, frogs aren't great for that. So that's really the advantages and disadvantages of using the Xenopus. Most of what we're going to look at with the Xenopus are commitment, looking at the cells in their specification process, when they become committed to the various germ fates and the germ lines, and then the cells in that regard. So that's what they're really good for. Fish models, or the zebrafish, is mostly used for ours. Lots and lots of embryos. They breed all year long. They're easy to go from one generation to the next. Zebrafish can be bred in the lab, and so they're good for generation after generation. Similar to the sea urchins, their embryos are transparent, so we can see a lot of the gastrulation processes that are occurring in the fish model. I showed you that video before, and I'll show it to you again, where it actually shows the time lapse of the embryo developing. You can see almost everything going on during those stages. So they're very great for looking at gastrulation. They develop outside of the mother, and the reason why I bring that up is because you'll see here in just a second regarding the mammals. That's one of the biggest disadvantages of mammals is it develops inside, and you can't really watch its development as you can with the fish models, with the Xenopus models. And then one of the big advantages, one of the things that we struggle with at times when we're doing genetic studies is integrating genomics into the cells. One of the nice things about this fish model is the cells are very permeable to some of the lipofectamines and other ways of introducing genetics, recombinant genetics into the cells. So they're great for fate maps, being able to tag them with GFP so we can look at where genes are being expressed. These are, so the fish model's been used substantially because of that, the Xenopus. Chickens. This is what I primarily deal with. I'm the chicken man. This regard, Toy Story. But we actually are doing chicken embryology for the cell biology lab, where we're going through the various stages, and we incubate them, and we're able to see them at the various stages throughout their whole development. Easy to raise outside. You can just constantly, we're getting eggs all semester long, and so we're able to breed them in the incubator. One of the great things about the chick model, or the chicken, is that we can time and know exactly what stage we want to look at just based upon temperature. All I have to do is put it at the right temperature and a certain number of hours, and boom, we know exactly where we want it to be when at development. So they're very predictable. Sometimes because of fluctuations in certain parameters in some of these models, you can't always time their development. There might be differences in some of the stages, but chickens are very easy to be like, hey, I want them at when their heart starts forming, like we did yesterday, and I know exactly how long to incubate them for that amount of time. One of the fascinating things about the chicken models is they're more similar in their earliest stages of gastrulation to humans than mice are. So especially in the earliest stages of movements in the formation of the 3 germ layers, chickens are almost identical in the formation of the 3 germ layers and how that's patterned as humans are, which is a huge advantage for us to be able to learn more about ourselves and using the chicken model. Another thing that's great about chickens is because they're in ovo, they're in the egg, we can not only do experimental transplantation, but we can also do genetic manipulation. They're very easy to manipulate genetically, and I did a number of genetic manipulation studies for my PhD, as well as some experimental manipulation. So they're great for both of these. You can manipulate them, cover them back up, and let them incubate longer. So you can just stop, do some things with them, let them keep growing, and then go look at them later on. They're absolutely wonderful which is why I chose them, for a model. I did have a choice in my PhD to use a mouse as a model, but I said screw that, because it takes months for you to get enough organisms to even do an experiment. And I had a guy on my committee. It took him 7 years to do his PhD. It took me 3.5. So what model you choose to study, I was able to do experiments with the chickens after 2 days, where it would be the equivalent of after 2 months with mice to get the same thing. So I chose wisely. Able to finish my PhD quickly. But he took 7 years in studying development using the mouse model. Now, mouse model does have advantages over all of the others. And a lot of the advantages are in terms of genetics and later development, they're the most similar to humans. So obviously we need to use them as a model because their genetics and their development past a certain point are so similar to us that how can we not? They're very easy to maintain within a housing environment, very easy to breed from one generation to the next. You can just keep these lines going of these mouse models back in most places except for here. we've always had a mouse facility where we've been able to breed them and do a number of genetic studies. Disadvantage, you don't get a lot, and it takes forever to get enough to work with to do some of these studies. You also don't get to manipulate them as easily as you do these other models because they're developing inside. Pretty much once you take them out, you're done. You can't put them back in. And a lot of the embryos are so small, it's not advantageous in terms of watching this. So there's several factors against the mouse model. But the trade-off, because of their similarity to our development in terms of genetics and later stage development, you can't ignore them. You have to use them as a developmental model to be able to understand us better. So these are all of the advantages and disadvantages of these models.